The dot plotting technique is a very simple method for detecting biological samples like proteins or nucleic acids. It is very similar in principle to the detection of samples in a western blot or a southern blot. In a dot blot assay, the sample of interest is not transblotted from a gel, but instead directly applied to or dotted on a membrane such as nitrocellulose or nylon. The sample is subsequently probed for detection. If the sample is directly labeled with a tag, the tag can be used for detection on the membrane. Biotin is a very commonly used label, although other labels are equally effective. The dot blot assay is a convenient and simple method to detect the efficiency of labeling of different probes. In this tutorial, we use the dot blot to determine the labeling efficiency of a sample with biotin by comparing its detection to known biotinylated reference standards. Make serial dilutions of both the test sample and also of the reference standard. Tenfold serial dilutions is a good place to start since it gives a wide range of sample concentrations. In other cases, fourfold or even twofold serial dilutions may be more informative. Be sure that the test sample and the reference standard are diluted in the same way. For a tenfold dilution series, aliquot 90 microliters of buffer in each tube. Take 10 microliters of the test sample and add it to the first tube with the buffer. Mix thoroughly and then take 10 microliters out of this tube and transfer it into the next one. Repeat this for the entire series. Make the reference standard series the same way. Prepare your membrane according to manufacturer specifications. Then take a small aliquot of the sample to dot onto the membrane. The dots should not be too big. Usually 1 microliter or even half a microliter is sufficient. Be sure that the dots are uniform in sample volume and dot size. Carefully dot the sample onto the membrane. Repeat this for all sample dilutions. Dot the serial dilutions of the reference standard in the same way adjacent to the test sample dots. Allow the dots to air dry over a couple of minutes. The membrane is now ready for detection. The label on the sample will determine the detection method. In this case, our biotin-labeled sample can be detected with any streptavidin or avidin enzyme conjugate, for example, streptavidin alkaline phosphatase or avidin horseradish peroxidase. A typical detection protocol is as follows. Incubate for 30 minutes in a blocking solution to minimize background, followed by 2 times 15-minute washes in TBST. Incubate for 30 minutes in an appropriate conjugate diluted in the blocking solution, followed by 2 times 15-minute washes in TBST. Finally, the membrane is transferred to the appropriate enzyme substrate for color development. This might be BCI-PNBT for alkaline phosphatase detection or DAB for peroxidase detection. Once the signal is developed, compare the intensity of the test sample dots to that of the reference standard. This comparison is a semi-quantitative way of determining the labeling efficiency of a test sample.